Kia ora and welcome to this Manaaki Whenua LINKOnline Webinar. I'm Christine Harper, Manaaki Whenua's Stakeholder Relationship Manager, and my job is helping to join the dots between our research and the questions that our stakeholders are grappling with. Wetlands have always featured strongly in our research. They sit uniquely at the intersection of land, water and biodiversity ecosystems and are complex, diverse and changing. Olivia Burge leads the Wetlands Research Program, and today she will outline current work on wetland vegetation measurements and touch on other work in the Wetlands Program that I'm sure you'll find interesting. I'll be back at the end to help with the questions, but don't forget to put them in the chat box as soon as you can. Over to you, Olivia.
Kia ora. Good morning, everyone. So I'm going to be talking about some of the work that we have been doing with regard to wetlands, particularly with a focus on nutrients, and some of the work we will be doing. Now, you'll have to forgive me, I've got a bit of a post-viral cough at the moment, and it's only bad when I talk and when it's cold. Unfortunately, we're 2 for 2 today. And I was going to put photos of everyone who's contributed to the work I'm talking about today on the screen, but there's far too many people, and I was having anxiety about leaving people off. But certainly some of the 3 key people have influenced my thinking in this area. We've got Bev Clarkson, who recently won the Loader Cup there. Hugh Robertson, Bev's at Manaaki Whenua too. We've got Hugh Robertson at Department of Conservation, who runs the Arawai Kakariki program, so DOC's Wetland Restoration Research Program, and Chris Tanner, one of the wetland experts at NIWA as well. Excuse me. So nutrients are really important with respect to wetlands. Now, I know it's typical to think about hydrology, which is also crucial because wetlands are wet places. But nutrients are super important too, and I hope to convince you of that a bit today. Empedisma and Sporodanthus are 2 of our key wetland plant species, for example, so they're native plants, and they're adapted to low nutrient environments. Some of the weeds on that list that you see there are able to exploit high nutrients and move in. So most of those species are exotic, except for raupo or taifa, which can become locally invasive where nutrients are high and has posed some problems for restoration projects where they would like more of a diverse mix within their wetland. And when I've got reflect changes in the wider environment there, well, lowland wetlands in particular are effectively the receiving environment for what's going on upstream in their catchment. So if a lot of nutrients are coming into the wetland, that tells us not just about we've got some things to think about in the wetland, but also about our land management upstream, both for the rivers or other water bodies that it flows down into the wetland, and whether we're doing best practice. Finally, in terms of the maps on in my slide there, we've lost about 90% of our pre-human wetlands in New Zealand. So it's critical to understand the key factors that affect wetland hydrology, like nutrients, in order to protect and conserve the wetlands that we do have left. So it's not all about anthropogenic nutrients though, so we need to do a little bit of increasing our baseline knowledge about wetland types, because wetland nutrients do vary by wetland type. If you think about, and this is a gross simplification, and I apologize to you who have a more nuanced understanding, if you think about 4 of our key wetland types, so bogs, fens, swamps and marshes, you can think of them as a bit of a gradient. And what we see along that gradient is that the water source varies by wetland type. So bogs are rainfall fed. Swamps and marshes at the other end of the gradient are groundwater and surface water fed. So immediately, if you think about where nutrients come from and how they're carried, you will expect that, for example, swamps and nutrients just naturally will be receiving through surface and groundwater, perhaps some of the nutrients from up catchment that are washed through. There are also some differences in terms of water flow and fluctuation, which can have an effect on the amount of nutrients coming in, and the pH, which can affect nutrient availability, but we won't go into today. As such, and I sort of already foreshadowed this, there's a natural gradient of nutrients where it's low in bogs. Fens are sort of the middle ground, and then higher in swamps and marshes. And for those of you who are interested in learning a bit more about this, I can really recommend Johnson and Gerbo's Wetland Types in New Zealand for a fuller discussion of this. And then if you're interested in how some of the transitions between these wetland types occur and how important it is to understand them, then I recommend my colleague Matt McGlone's paper up there. So we know that wetland soil nutrients can differ naturally, but all can be affected by anthropogenic, so that's human input. And this is despite the fact that we often talk or hear wetlands talked about as the kidneys of the ecosystem, so filtering out nutrients. So this is true, but particularly in systems that are low nutrient adapted, this can go far too far. If you think about where anthropogenic input might come from, surface water flow, aerial deposition, and then drains, particularly when they run through the middle of a wetland, because you would think drains take water away from a wetland, but actually if there's a big flood event, they can carry water into nutrient sediment-rich water into the wetland as well during those events. So there's a reference down there to a thesis if you're interested. And before I go too far, I added this in this morning. I'm primarily talking about nitrogen and phosphorus today, and people often refer to those as N and P, and I probably will as well. And so that's what they mean. I thought we should get that out of the way early. Some of the key questions that we talk about, particularly with regional councils, so we've partnered with a few in terms of doing this work, which has been, we've had such stimulating and interesting discussions, and they've really helped steer our work. How to detect ecologically relevant anthropogenic input. Can I just say first off, so total N and total P from soil, so taking soil samples, are great. They do include locked up nitrogen and phosphorus that isn't so plant available, so it's less potentially ecologically relevant, at least in the short term. And as such, any recent additions of nitrogen and phosphorus that are plant available will be diluted to some extent. However, after, for example, fertilization experiments, we do see that nitrogen and phosphorus, as reflected in the total soil NMP, does increase as we expect. So I'm not knocking it. It is it's fairly expensive as a test compared to, say, monitoring the foliage, which is what I'll come on to. And also, it's technically more difficult to take a soil sample than to take a foliage sample, so it's the training and the time that's involved too. Plant available soil NMP, we can test for that too. That's a more technical test, and then you're adding on another test in terms of your budget. But to do this, to really monitor this well, this is not something you would do once every 5 years. So that's more intensive monitoring for sites where you've got real concerns, and you would have multiple spatial points of monitoring and then multiple samples potentially each year. So it's not really excuse me, suitable for widespread monitoring. But as I mentioned, foliage is easier to sample, so it's more cost effective. And we're really getting at, because of the issue about whether how much of a sample is actually plant available, we're really getting at what's relevant to the plants. We're letting the plants tell us what's going on. So you can sort of think about it as blood tests for plants. We often do this with people. So we take iron, for example, samples, and then we can see of the food that you've been eating, we can infer how much iron was in it and also how well you're digesting it. And so the plants are effectively acting as bioindicators. So we've done some research taking the idea about foliage samples and how useful they are to monitor wetlands and address two questions. So the first is, can we predict which nutrient of NMP a wetland is more likely to respond to. So, and that's known as which nutrient is more limiting. And that's using the N to P ratio. And secondly, can we correlate foliar NMP to tell us what the soil NMP, the total NMP, is doing, and potentially at least sometimes only do the foliage monitoring to save on monitoring costs. So I'll address question 1 first. So when we're looking at which nutrient will have the most effect ecologically, we take the foliar N to P ratio within the plant. What this means is we're not looking at how much total nitrogen is in the foliage, nor are we looking at the total phosphorus in the foliage, but the ratio between the two. So it's a relative amount. Just divide N by P. We'll calculate it. And then if you're really looking to test this though, to test whether this N to P ratio is telling you which element is limiting, then you need to fertilize with both nitrogen and phosphorus. Early work in the 1990s established this as a potential indicator for wetlands. There has been some discussion about the exact ratio, so the cutoff between N and P limitation, and maybe where there's co-limitation. It's all interesting stuff. But a lot of that work was done in the Northern Hemisphere, which obviously has different plant communities, different levels of atmospheric nitrogen input, and quite highly managed wetlands in terms of where that work was done. So recent work by all three organisations, so we're talking DOC, NIWA and Manaaki Whenua, tested this in Otu Wharikai, so Ashburton Lakes Wetland Complex. We took foliage samples at pre-fertilisation, and this suggested that there was N limitation. So that means that if you add nitrogen and phosphorus, nitrogen should provoke an effect. And I should back up a little bit. When we say an effect, what we mean is, will the vegetation community increase in biomass? Just like if you fertilize your plants, or your crops, you're expecting to see an increase in biomass. We added what we'd call realistic farm runoff levels of nitrogen and phosphorus. So there's also an element of understanding the ecology of farms and wetlands nearby. And this was added for 4 years. And then after 4 years, the above ground biomass, so no roots, was harvested. So we had nitrogen, phosphorus, and both, and a control where it was unfertilized. And what we saw was, with our highest level of N, we saw quite a substantial increase in biomass, so 28%, and no biomass response to P or phosphorus. And so this supports the prediction by the foliage N to P ratio that it was nitrogen that was limiting the community. Other interesting things occurred, but you can read the paper which was on the previous slide if you want to find out. So going a bit further, because that's quite a rough which element is more limiting, in discussion with regional councils about the pragmatics of monitoring, we investigated the correlation between foliage N and soil N, and foliage P and soil P. And if you think about it, particularly in terms of like the iron in the food you eat, you're not going to expect a one-to-one relationship, right? And the second thing to note is that there's a well-documented phenomenon where plants will take up phosphorus even when they don't need it. And so that's called luxury phosphorus uptake. That's actually really great for monitoring purposes. And so we expected between the foliage of plants and the soil, a stronger relationship between foliar P and soil P compared to nitrogen. We used the New Zealand wetland database supplemented by data from councils, and that gave us over 1,600 foliar samples. And then we did a single species analysis as a first step. And so we had 12 species that we were comfortable. We had sufficient replicates to do this. And this is freely available online. So if you want more details, please do go look it up. This is not the kind of relationship you want to see if you are thinking about substituting solely foliar monitoring for soil monitoring. So what we see is, firstly, the red line, which is the one-to-one line. As I said, that's not super important, but it's just to help visually orientate you within the graph. So 2 species here on the panels. So Manuka there and Nakaraina. And as you see, as for both panels, as soil nitrogen increases, there's actually very little response in terms of the foliage. It's almost flat. Even if that was a relationship you could work with, the spread of the data, which you can see there, is such that we can't use this. For those who are interested, this is actually an orthogonal regression, so we're assuming errors in both X and Y. In terms of phosphorus, it was better, as we'd sort of expected, but it's useful to understand, right? Like you can see, particularly with Manuka there, that we would expect to see as higher values for foliage as soil increases. But again, in terms of substituting it for monitoring, when you've got this kind of spread in the data, you still have some concerns. In terms of, I'll just take a sip of tea before I get excited about the future work. Future work to go a bit further. In terms of species selection, so which species we're monitoring, dominance has been linked, so very abundant species, has been linked to very conservative nutrient uptake. Species that have very conservative nutrient uptake will be shockingly bad as bioindicators. But we actually, we do our foliage monitoring from the dominant species. And so I think there's a real interesting question there about, now pragmatics to be addressed, but can some of the less abundant plant species in the community tell us a bit more? There's also the H index. For scholars among you, there's not how many papers that a plant publishes, but the H index tells us, as I was saying, how conservative at the species level a species is with respect to nutrient uptake. And if we have permanent plots, the theory suggests that as nutrients increase, the average H index in the community will also change. And so that's quite useful because once you've determined the H index for a species, there we are. You can, at some point, you can actually cease foliage monitoring for these purposes because you have a library and you've calculated those H indexes. I should say the H index is really just how steep that slope is that you saw on those panel graphs previously. So you do that both for nitrogen and phosphorus. So to summarize where we're up to, on the basis of one site only, we can see a promise that we can adopt the international approach of predict whether nitrogen or phosphorus is more limiting using the foliar N to P ratio. But in terms of the correlation between foliar N and P and soil N and P, we can't really support that as a drop-in to only monitor foliar nutrients. I think with some of that future work that I discussed, we will certainly deepen our knowledge of plant nutrient relationships and so our understanding of the ecology of wetlands, even if the foliage remains not a great substitute, a direct substitute for soil monitoring. Finally, it is so tangentially, well, indirectly related. We're working on classifying wetland vegetation communities and then linking them to nutrients. What we actually expect to see is that the biomass response to fertilization, so as nutrients increase, comes from new species who come in and are able to exploit nutrients. That turnover can take a long time, so there can be a time lag. So in the Otuwharika experiment I discussed earlier, 4 years probably wasn't long enough, particularly given it was a modest fertilization. So what we want to ask, given the large amount of vegetation plot data we do have from wetlands, we think the time is right to ask, how do plant communities change along nutrient gradients? And just bearing in mind that this is a slight, well, I said it was indirectly related. This has not changed necessarily within a wetland. We're looking at multiple wetlands. So you might call that a space for time substitution. So a classification, which is what I just mentioned, clusters vegetation plot data into groupings. A classification has already been done for New Zealand's non-forested vegetation and the forest vegetation, but it only came up with two communities or groups that of wetlands for mainland New Zealand. This is because the plot data that was held in the New Zealand wetland database wasn't included. So we think this is actually a real big opportunity to expand this existing classification and do a quantitative classification of New Zealand wetland vegetation. And this is where it might differ to other classifications that you've heard of wetland vegetation. In terms of progress to date for this classification, we assessed where all our plot data was from, thanks to Robbie Price, one of my colleagues at work. And he calculated for us both where plots are, and you can see with the red circles, there's two big gaps, but there's other gaps on the map. So we actually looked at for the wetland area within, in this case, different lens environments, which areas were most undersampled, and we came up with these two. So earlier this year, we spent 2 weeks doing fieldwork in those two locations and added about 20 wetlands and numerous plots to our data set, which is really excellent. Some of the key questions we'll look at in terms of the vegetation classification is, do we have enough data, because it's a data-driven approach, to reflect New Zealand wetland diversity across wetland types? We know, for example, that marshes have been undersampled in terms of some of the data we do have. And also for me, some of these ecotones we don't see so regularly now. So in this image, you can see the forest grading into peat bog-like vegetation. Some of those have been lost through fire. So it'll be interesting for me personally to see which communities we do see and which we don't find, which is linked to what we see in the landscape today. And then we'll look to link to those groupings that we see in the classification to soil N and soil P. So to summarize, there's early work, at least in terms of New Zealand, that we can predict whether a wetland will be more vulnerable to change from nitrogen or phosphorus using the foliar N to P ratio. We can't yet directly correlate foliar N and P to soil N and P and use that to infer change or substitute it for monitoring. But there are exciting avenues to take this further in the future, and that at minimum will really increase our understanding of wetland ecology as that relates to nutrients. And finally, we are working, as I just covered off, to classify New Zealand's wetland vegetation, so a plot-based classification, and we'll seek to link that to the soil nutrient data that has been really assiduously collected by regional councils, Department of Conservation, and CRIs like Manaaki Whenua and Niwa. So thank you all.
Thanks very much, Olivia. We've got a number of questions coming in, and we're not going to get to deal with them all today. And we're going to be kind to you as well, because we can hear that this is a big effort for you to talk to us today. So thank you very much for that. But there are a few, so let me just start from the top, and I'm going to start with my question, which is, what is your favourite wetland plant?
So I find the carnivorous plants totally fascinating, right? So these are the ones that eat little insects, but I would say it's probably, especially at the moment with respect to this work, Empedisma. So Empedisma, as I mentioned at the start, is really adapted to low nutrients, and it's got a special form of roots called cluster roots, and this allows it to really be super effective at low nutrient levels. These are also, the cluster roots is also what is quite an important contributor to the peat formation that we see in bogs. And we also understand that cluster roots, because they're not as needed in terms of high nutrients, so they decrease in abundance sometimes. And so I'm really interested to understand a bit more whether in higher nutrient situations, we've actually got peak formation slowing down because of that reduction in cluster roots. It's a fascinating, fascinating species. There's two. There's some work, I can chuck a link in the update if anyone's interested about a little bit more about the Emphysema species of New Zealand.
Fantastic. And that leads really neatly into this next question that Owen has asked, which is it worth trying to obtain a soil sample from the root mass area? And he was thinking about that in Raupo that is growing out over open water, but maybe it's a general question as well.
Oh, yeah, good question. One of the things, so I guess you're talking probably about the smaller samples. So what we do is we tend to take a core sample, which I don't know, you may or may not be able to see my hands, is about yay big. And that allows us to sample for bulk density. So we just do 2 big cores like that. Another common practice is to take 4 small cores and mix them up. So you're incorporating perhaps a little bit of the spatial variability within a plot. It's not like it's not a terrible idea in terms of, I guess you're really talking about targeting the soil in which those roots are growing, but it could be shockingly difficult with the cores we use. So some of what I was discussing is some of the practicalities with getting a soil core. So we know this from even just from our field work recently in quite difficult locations. And so that might be because it's super sloppy and you've just got it running out of the core. Or you don't want to force it down the core too much because you can compact the soil, which affects your bulk density estimates. And so if you're trying to push it through roots, you've got some issues there too. So we rely on the assumption that the cores that we take, which are in the southwest corner of the plot, reflect the nutrients available to vegetation within that plot, despite the fact there will be some micro scale, potentially topography effects or small scale effects on nutrients. I think Greater Wellington Regional Council has done multiple cores though for plots, and we're fairly happy with how I think this is correct, I don't want to speak too much, and I think they were fairly happy with how that turned out though.
One last question. What seasons were your samples taken? And is NNP correlated with temperature or soil moisture?
Yeah, so I'm going to assume this is in the foliage. So what we recommend to take to do the samples during the growing season, and there's some quite specific. So there's the Wetland Monitoring Handbook, lead authored by Bev Clarkson at Manaaki Whenua, and that's got some more specifics about when to take samples. Variation in plant foliage, this is, so I was talking about soil before, can be quite high. So within species variation within a site where you assume that it's got the same level of nutrients coming in can be surprisingly high. So that's why, to try and control at least that variation, we suggest taking from multiple individuals to try and average out across that. We haven't directly tested climate or what was the other one? Moisture. But certainly because you'll have plant composition turnover with respect to those environmental factors. So I don't know within a species exactly how that would work, but you would expect to see compositional turnover, and therefore for the same given soil nutrients, what I was talking about before, that conservatism, you might see that change, and therefore a change in the foliage for that plot, even though it's a different species.
Fantastic. Look, I can see there are some amazingly interesting and quite technical questions coming through. So we'll put those.
Thank you for your interest, everyone.
We'll put those to Olivia. Thanks heaps, Olivia, for getting through this. Fantastic presentation, and watch this space for our next LINK webinar later this month. Thanks.
Bye for now.